Okay, so thank you very much for spending your time to spending your time. So it's my great honor to be here to share with you my experience about MRI. I'm sure the audience here may have much more experience about brain mapping and molecular imaging. That's true because I came from a background of technical development, and I started my scientific career by building NMR probes, following the same trajectory that my thesis advisor did more than half a century ago here in a research laboratory of electronics at MIT. So this was a very good experience, very good way to start building a manshine from the very fundamentals. And that sometimes will lead to revolutionary breakthroughs. So my thesis advisor actually did it, made a breakthrough, made an important breakthrough 46 years ago. But when we shared about the awarding of the Nobel Prizes to the NMR field in 2002 and 2003, my thesis advisor held a cigar and shared with us that the continuous awarding of 2 big prizes in 2 consecutive years means that the technical development of NMR might not be so fruitful in the future. And in fact, he actually gave an acronym of NMR, which means no more research. But at the same time, he also commented that although we students had eventually focused ourselves on practical applications of NMR, we should always bear in mind the fundamental challenges that still remain ahead of us. And we should cherish those ideas that randomly generated in the lab as pine nuts coming out of the jar, because his last name is also Pines. Okay, so, and we cherish those seeds and hoping that sometimes it might flourish into a forest, hopefully. So throughout all these years, I have been focused on the technical development of alternative detection technologies to improve the applicability and the sensitivity of NMR. And I have been very much indebted to these brilliant world-class researchers who shared with me their brilliant insights. These are the pictures. These are the pictures when they are young. And I'm sure that some of you might know of. And before the phased array technology became dominant, most of these brilliant scientists have benefited in one way or the other, the technical development of microcoils. The initial development of microcoils started in the early 1990s. And because microcoils have a higher sensitivity, the higher sensitivity came from the fact that the same coil that is placed far away from the object would detect the same amount of magnetic flux as a smaller coil that's placed closer to the detection object. The smaller coil will detect the same signal, but the less noise, according to the signal-to-noise equation, which is the fundamental basics of NMR sensitivity. And then, if we have a way to insert this coil into the body cavities, and if we have a way to insert them into the blood vessel, for example, this smaller coil will be very important as a tracer to track the position of the catheter for interventional procedures. Alternatively, they can also be used as an interventional coils to detect lesions outside the lumens, for example, to detect prostate tumors from the rectums. So it would be ideal if this kind of coils are not connected to any kind of metallic structures, because as we all know, metallic wires are good antennas, which might introduce unpredictable burning or heating inside MRI scanner. In principle, we could transmit NMR signal wirelessly by inductive coupling. But in practice, this kind of wireless coupling would introduce tremendous amount of signal loss. So whether there is a way for us to incorporate an amplifier on board on the small coil, and to amplify the NMR signal in situ before these signals attenuate. In this way, we can eliminate the burden, the cumbersome conductor wires, and at the same time, we can also maintain the superior local sensitivity of the local coils. So the search for a wireless powered NMR amplifiers and the miniaturization of these wireless amplifiers on board and to pack them into compact space is my continuous work. Okay, so besides NMR, in the world of sensing technology, similar requirement also exists. For example, for a pressure sensor that operates inside the bladder, for example, the pressure sensor can instantaneously tell the external receiver what's the pressure inside the bladder. And this kind of continuous sensor will be very important for patients with incontinency. Because the sensor will operate inside the body cavities for extended period of time, and we would want the sensor to be batteryless and wireless. So, and again, in principle, this kind of sensors can be measured wirelessly because the pressure change inside the bladder can be transduced to capacitance change of the sensor. And this capacitance change can also be read wirelessly by its transmission frequency change. But in practice, because the sensor is deeply embedded inside the body, the power transmission curve will be very inefficient. So this is the ideal case where the pressure-induced frequency change can be easily identified if the sensor is shallow enough. And this is the reality case. When the sensor is deep enough, we can really read nothing from the external network analyzer. So it will be ideal if we can use the same idea of wireless pumping to charge up the sensor in situ, so that the sensor can emit its own signal that's dependent on its local pressure. So we have two questions. One is in situ NMR signal amplification. The other is in situ sensor signal amplification. And then they can both be implemented by the same technology, which we called the wireless powered amplifier. The wireless powered amplifier doesn't require any DC power to operate. Instead, it can directly convert the external RF power into amplified signals. So this is a little bit more detail about operation principles. Suppose we have a weak MR signal that's at the frequency of omega S. And suppose we have a strong external pumping signal at omega P. And suppose if the device can perform some nonlinear interaction to enable the weak MR signal to exchange energy with the external pumping signal. After energy exchange, they will generate the signal at a different frequency, which we called the idler signal. We called it the idler signal because this signal is not directly detected. Instead, it will mix back with the pumping signal and generate a secondary amplified signal at the original MR frequency. So the net result is, after multiple stage nonlinear interaction, the MR signal is amplified by an RF signal and generate an amplified output at the same LAMO frequency. Because this sensor can detect MR signal and amplify MR signal, and potentially be integrated with other types of physiological sensors to monitor physiological parameters in situ. So this multimodal, multi-purpose sensor can perform multiple functions. And then we give it a magic name called wand, which is called magic wand, magic rod. And this is a work I collaborated with Dr. Yu in 2002, and we published a paper in Radiology. So here's the detailed circuitry of the sensor. So the nonlinear circuit, the first generation circuit has 3 meshes. It's basically a triple frequency resonator. So with one of its capacitors substituted by a nonlinear capacitor, which we call the varactor. And the resonator also couples with a receiving loop, which is just an ordinary NMR surface coil. And it also inductively coupled with the pumping loop, which will provide the wireless energy to charge up the sensor. So the sensor has 3 resonance frequencies. By making the sum of the 2 lower resonance frequency approximately equal to its higher resonance frequency, efficient signal amplification can occur. This amplification derives from our ability to wirelessly, to periodically manipulate the capacitance. The time-dependent capacitance is described by this equation where the M is the modulation factor. So it's important to mention that the operating frequency of the sensor, omega 1, omega 2, and omega 3, are not necessarily equal to the resonance frequency of the sensor. So this is because when we make the sensor and embed them into the body cavities, so we can no longer retune it. But this is not a problem because as long as we can satisfy a certain relation to make the off-resonant effect at one resonance frequency to be equal to the off-resonant effect at the second resonance frequency, then we can effectively make the sensor to perform efficiently at the particular operating frequency. This is what we call the frequency matching condition. And by fine-tune the pumping signal to make the modulation amplitude, the square of the modulation amplitude multiplied by the qualitative factors equal to 1 plus kappa square, the efficient oscillation can occur. So I should also mention that if the pumping signal is strong enough, the sensor is an oscillator that will spontaneously emit sustained oscillation signal. The sensor will be used to detect physiological signals, even outside the NMR scanner. But when the oscillation signal is small enough, not sufficient, we operate the sensor as an MRI amplifier. And the amplification factor, the gain, is equal to square of the inverse of the relative derivation from the oscillation condition. So this is a little bit complex, but fortunately, we derived the equation and get to know this detailed relation, this property. So the net result is that we can wirelessly tune, and we can wirelessly tune its resonance frequency, and we can wirelessly tune its gain and amplitude. So this is a very good feature to operate inside body cavities. So as a first generation device, we actually did some simplification of the circuit. We simplified the triple frequency resonator into a double frequency resonator because we let the lower 2 resonance frequencies share the same resonance mode. And this is a device that I built from off-the-shelf components, and that is coated by PDMS, which is bio-inert and biocompatible. And then we use a pair of loop antennas connected to a network analyzer to analyze its power response, this frequency response curve. As we can see in this plot, when the sensor is in its passive mode, so its center frequency is about 496 megahertz. But as we crank up the pumping power, the sensor center frequency gradually shift to 500 megahertz with the increased height of its power transmission curve. So that's to say that we can wirelessly tune the off-resonance sensor to operate at 500 megahertz, which is the normal frequency of a 11.7 Tesla scanner. So inside the scanner, we put a sensor on a water phantom and separate the sensor from the external receiving coil, the surface coil, by a certain distance. So this is to emulate the condition that the sensor is deeply buried inside the body and separated from the external detector. And then we turn on the excitation pulse, which is the B1 transmit pulse. Because the sensor itself has a diode, the strong excitation pulse will detune the diode and detune the sensor so that we can wirelessly decouple the sensor from the transmit pulse. And then during the receiving stage, we turn on the pumping loop to pump RF energy into the sensor. The sensor will re-emit the backscattered signal and amplify the signal for the external receiver to detect. So this is the operation principle. And then, as I have mentioned earlier, the sensor has the intrinsic circuitry to detune from the transmit pulse. So as an initial demonstration for in vivo application, we first did implantation. So I will mention later that we will remove the implantation. So in our first implantation experiment, we implant the sensor inside the medial surface of the kidney, inside the large rat. We chose this surface because this surface is far away from the body, and then it was traditionally difficult to detect at very high resolution for MR microscopy. And in the lower resolution image, we can see the kidney layers, but we don't see the fine structures of the kidney. But as we turn on the amplifier and obtain higher resolution image at 70 micron in plane resolution, we can see there are multiple black spots on the renal cortex. And these black spots are particularly obvious in the zoomed in view that's defined by the green dashed box. So of course we get these black spots because we also injected a contrast agent called catenized ferritin. So based on our previous histology study, catenized ferritin will selectively bind to the membrane, to the glomeruli basement membrane. And then it will stick to the glomeruli to make the glomeruli signal look dimmer. So this is the first step demonstration of the high resolution capability of the amplified NMR detector. And then we ask the question whether we can see renal microstructures without the presence of contrast agent. We also succeeded. So in this T1 weighted image, we can actually visualize multiple bright spots in the renal cortex. And these bright spots may come from the increased water relaxation, or it may come from blood flow. So in order to exclude one possibility or the other, we apply saturation pulse, and we found that the bright spots completely disappear. So this made us conclude that the bright spots correspond to the higher blood flow. And then we inject fettanized keratin and obtain T2 weighted image. We observe also multiple black spots that with very similar pattern of the bright spots before contrast agent injection. So this made us conclude that we can actually utilize the blood flow inside the glomeruli to visualize glomeruli themselves and without the need for contrast agent injection. Because most contrast agent will perturb the renal function to a certain extent and is secreted by its kidney. So the ability to non-invasively and non-invasively visualize glomeruli distribution in the absence of contrast agent will be a plus to study renal microphysiology. Despite of our interest in visualizing kidney microstructures without contrast agent, we are actually not opposed to the use of contrast agents, especially if these contrast agents can emulate some cations that are important for biological function. For example, manganese is extensively used as emulator for calcium to study calcium metabolism. So after injection of manganese, we again see multiple black spots or dimmer spots in the renal cortex. We suspect they correspond to glomeruli as well. And then we did co-registration experiment and to visualize the bright spots before contrast agent administration. And we also see the correlated pattern before and after contrast agent administration. So this made us conclude that manganese can also visualize glomeruli. But because manganese is a positive contrast agent, and because manganese will be absorbed by the renal epithelial, by the renal tubule epithelial cells, we hypothesize that manganese will increase the signal content, the signal intensity of renal tubules to a larger extent than glomeruli. And we test this hypothesis by a time-dependent experiment. So in this experiment, we apply saturation pulses before manganese injection to completely suppress contrast, endogenous contrast introduced by blood flow, and we gradually increased manganese infusion amount, volume, until there are multiple white bundles appearing in the renal cortex. And then we stop the injection, wait for about 40 minutes. We saw that there are multiple black spots appearing, and the pattern became more and more clearer. If we plot the relative intensity of each pixel with respect to its pre-contrast image, we can find that most of the pixels in the bright region, which is defined by green, they have higher intensities than the pixels in the dimmer region. So basically, both dimmer region and the bright region, they have increased signal intensity, but the bright region increased to a larger extent. So the relative ratio between the signal intensity increase in the brighter region and the dimmer region can tell us something about the differential absorption of manganese in the renal tubules versus glomeruli. So this kind of high resolution capability will enable us to study the microscopic mechanism of contrast agent distribution. So even though molecular imaging favors the use of lower resolution images, so our wireless amplifier will tell us better about how molecular contrast agents works. Okay, so it's great to have the implantable detector, but in the following, I want to explore the possibility of using the wireless detector in a non-surgical way. So we have many body cavities like esophagus and rectum, for example, the digestive tract. So whether it's possible for us to insert a detector instead of implanting a detector, we insert the detector into the digestive tract and observe something interesting around the digestive tract. So this is our hypothesis. So in order to realize this goal, we have to build cylindrical detectors that are easier to insert. So in this diagram, I have shown one example of the cylindrical detector that can work similarly better as an implantable detector. In this cylindrical detector, it has two identical vertical legs that are interlaced by end rings. So the end ring, each end ring has a pair of varactor diodes that are aligned head to tail configuration. So once we turn on the pumping field on the actually oriented pumping coil, the pumping field will modulate the capacitance of the diodes on the end ring, and in order to achieve efficient energy transfer from the pumping field to the MR signal. And then the MR signal will be received by the external receiver. So we did some SAR simulation in CST Studio, assuming that the required pumping power is about 0.6 dB lower than the expected. And then we figured out that the detector has only non-negligible SAR around its end ring varactors. So even this non-negligible SAR is only 0.64 watt per kilograms, which is far below the average power dissipation limited by the IEC. So this means that we can actually safely operate the detector, and we will only get about 0.61 millekilocalvin per second temperature rise. So this is non-negligible. This is negligible. This is non-detectable by ordinary temperature probes. And then as an initial demonstration of the insertable detector, we insert this into the esophagus and observe the ascending arteries from the aortic arch. We chose this region because of its potential clinical relevance, because plaques in the aortic arch and in the ascending arteries are difficult to observe. But these plaques cannot be neglected because they are potential risk factors for ischemic strokes. So as we can see in this non-amplified image, because of the closeness of the detector to the aortic arch, only signals near the aortic arch are observable. But if we turn on the amplifier, and then we get at least another 3-fold increase in the detection sensitivity of aorta walls and in the walls of the ascending arteries. We can clearly see the artery walls and the fissure structures. So this kind of detector can potentially offer us at least a research tool to study atherosclerosis in the deep-lying arteries. But if we go back to our original kidney application, in order to use this kind of internal detectors to detect kidneys, we can probably figure out an alternative insertion pathway from the rectum. And then after we insert the detector into the rectum, when the detector is close to the kidney, we can clearly observe multiple bright spots on the surface of renal cortex. So this means after several years of effort, we have successfully removed the implantable detector and then used the insertable detector to detect kidney microstructures as well. As one further step, we also acquire the longitudinal view of the kidney to get a better view of the better overall view of the kidney. So without amplification, we get images. We get images that can clearly visualize overall kidney layers. But because of the relative insensitivity of external detection, we don't get very clear view of the internal structures of each layer, like outer medulla, inner medulla, etc. We only see some slight structures in the upper portion of the kidney. This upper portion of kidney is close to the passive detector in its unamplified state. This passive detector will give us about 3-fold increase in sensitivity when compared to the bottom portion of the image that is detected by the external coil alone. So after we turn on the amplifier, we can see another 3-fold increase in sensitivity. So basically, compared to passive detection, the amplified detector will give us 9 to 10-fold increase in detection sensitivity. And then we can see many white bundles in the renal medulla. And these white bundles correspond to medullary rays. Medullary rays are vasodilata that are interlaced with their adjacent space supplied by capillary plexus. So the approximate distance between medullary rays is about 0.2 millimeter. Okay, so, and then we also did some functional bone imaging on the kidney. So we first exposed the rat in 50% breathing, oxygen breathing condition. And then we can see multiple black spots in the renal cortex. After we switch the breathing condition from 50% to 96%, we can see that most of the black spots disappear. And the black spots increased in signal intensity by a factor of 2, but white bundles in the renal medulla increase in signal intensity by a factor of 1.4. This indicates to us that regions that were originally hypointensity will have a higher potential to increase in their intensity. So in order to quantify the differential hemodynamic responses of individual pixels in the renal cortex, we calculate the medium hypoxia ratio of each pixel. The medium hypoxia ratio is defined by the pixel intensity under medium breathing condition divided by the pixel intensity under hypoxia breathing condition. And in order to identify the local image contrast of each pixel under the 50% oxygen breathing condition, we also calculate the relative intensity of each pixel with respect to the local average. And then we plot the medium hypoxia ratio with respect to the relative intensity and find a positive correlation between these two variables. This positive correlation can probably be attributed to the proportionality between oxygen-bearing capability and the local levels of the oxygen hemoglobin. But of course, further study is needed to better understand this positive correlation. So although both MRI has already been used to map the overall layers of oxygen levels in kidney, due to the relatively low detection sensitivity of external detectors, conventional bone MRI cannot have enough spatial resolution to discern differential hemodynamic response of kidney microstructures. With the enhanced detection sensitivity delivered by the wireless detector, it's possible for us to individually identify the differential bone responses of renal microstructures, which are closely related to local changes of oxygenation. Because renal hypoxia is a common etiological factor that starts from localized region, the ability to identify distinct hemodynamic responses of renal microstructures will pave way for deep understanding of early stages of kidney diseases, at least in animal models. So next, I want to shift gear a little bit about the wireless detector that operates in the absence of MR scanner. So in order to operate the wireless detector in the absence of MR scanner, we want them to emit signals spontaneously. As we mentioned previously, we want the sensors to operate in its oscillation mode. So in this diagram, I have listed the circuit diagram of a sensor that we are currently using to measure the pressure. So this sensor consists of two overlaid circuits. So each circuit is a square circuit that have one square and two capacitors or two diodes. So when these two circuits overlay, they have stability, they have inductive coupling. The coupling strength will modulate their resonance frequency. So if we can read out the resonance frequency wirelessly, then we can translate to the pressure applied on the substrate. So in addition to the dipole mode that are both present in each sensor, in each resonator, the parametric oscillator also have an additional mode, which is called the butterfly mode. So the butterfly mode is created by bridging the virtual zero voltage points on the circuitry to create an additional mode. And then when the dipole antenna provide the sufficiently strong magnetic field, it will modulate the capacitor to create oscillating current in both modes. And then this is a picture of the sensor, of the overlaid sensor that are sandwiched by a polyurethane foam, which is sensitive to the pressure changes as a format of deformation. And then these are the two loop antennas to detect a signal emitted from the sensor. So as we can see from this image, from this plots, in the absence of amplification, so we can virtually detect no signals from the sensor when the distance separation from the sensor is about 5-fold the sensor's own dimension. But once we turn on the pumping power, the sensor's oscillation signal shows up as sharp peak in the frequency response curve. As we increase the pressure applied on the sensor, we can see the sensitive change of the frequency response curve. So then we plot the frequency response with respect to the applied pressure, and then we get a linear response. So although the sensor will have slightly different intersection with respect to the plot curve, so their slope remains the same. Every change in 1 Pascal will result in a shift of about 1.1 kilohertz. So this is very sensitive because the 1.1 kilohertz is much larger than the resonance frequency curve, the peak width of the resonance frequency curve. So as a rough estimate, we estimate the sensor to be able to reliably measure pressure change as small as 27 Pascal. So this is already approaching what can be achieved by wired technology. So as I mentioned earlier, because these sensors have varactors, we can also incorporate electrodes across the varactors to change the resonance frequency. So in this slide, I also measure the voltage-dependent linear frequency shift. Without amplification, we see nothing. And with amplification, we see very sensitive change of the frequency with respect to very small voltage change. So every 1 millivolts increase in voltage would introduce about 15-fold kilohertz shift in resonance frequency. So this is much larger than the peak width itself. And then if we want to measure the voltage change in the time domain, we actually use the oscilloscope to measure the oscillation frequency. And then after we mix down the signal and then deconvolute it, FM demodulated by a MATLAB, we can actually recover the voltage waveforms that are directly applied on the electrodes. So as we can see in this series of plots, the sensor have very good linear response from 73 microvolts to 9.0 millivolts. That's very large linear response range. And then based on the estimation of the noise level in the plateau region, the noise level corresponds to a noise voltage of only about 4.7 millivolts, which is much smaller than the neuronal spikes. So the sensor, right now we are sensing it, we are sampling it at 625 kilohertz. So if we sample it at faster rate, we can increase the dynamic range. So the 625 kilohertz will give us the maximum detectable voltage about 20 millivolts before it saturates. But if we sample at faster rate, Bruker can sample at 1 megahertz. So we can increase the linear range by a factor of 2. So which is much larger than neuronal voltage. Okay, at least for the local field potential applications. So next, I want to ask whether we can use the sensor both as an electrode, wireless electrode, and as an MR detector, so that we can correlate electrophysiology with MR detection. As an initial step, we use the MR detector and wirelessly couple the detector with human clinical coils. This will provide us with additional flexibility to use clinical scanners that are normally available in many hospitals and to use it for animal studies. So in this preliminary study, we used the clinical scanner to scan the brain of a small mice. That's very large size mismatch. But again, within a reasonable amount of time, we get high resolution images in both the transverse and the longitudinal images. We can clearly see the brain layers and the vascular structures in the brain. So in the next step, which I will also work extensively with Qian in the future, we will incorporate these electrodes into the sensors to record the electrode signal with the MRI scanners. And then we also want to build array detectors. So in MR applications, we often have the expectation to compare signal from the left side or right side of the brain. So that means we want to switch detection mode from simultaneous detection to selective detection. So in order to implement this, we can make the sensor, each sensor, responsive to several pumping frequencies. For example, this sensor labeled by pink, it has 2 butterfly modes and 1 dipole modes. The butterfly modes resonate at 225 and 500, and the dipole mode resonate at 300. So if we pump the pink resonator at 525, it will excite the pink resonator, because the cyan resonator also operate at 525. So if we pump at 525, we can actually excite them together and amplify them together. So this is our preliminary image with amplified signals from both sides. But if we pump only at the 800 megahertz, we will only excite the left sensor. If we pump at 900 megahertz, we will only excite the right sensor. And then in principle, we can also do it multiple times and get multi-slice selection. We can do selective left side or right side image over different slices. Okay, so eventually we want to apply this technology to a project that I have been working with Qian in the past and will continue work in the future. So we apply saturation pulses. Then we only detect a very narrow region in the brain. Then we remove the phase encoding gradient to accelerate the imaging speed. We image each slice at a repetition rate of 50 milliseconds. And then, for example, in this horizontal 1D profile, every horizontal line corresponds to the 1D signal profile at a particular time point. And then we observe the delayed onset response after 4-pole stimulation. But as we can see in this figure, the signal quality for lower averages is pretty poor. That is because of the lower sensitivity of the conventional external MR detectors. After multiple averaging, we get better signal, but we also lose the information about the low frequency fluctuation of the brain. So it will be ideal if we can use the wireless detector array to do the line scanning, and then we get better signal with lower averages in order to catch up with the low frequency fluctuation that's most important for resting state MRI. So in in conclusion, we started from the intuitive intention to increase the weak biological signals in vivo. And then we used the nonlinear circuits to make MRI amplifiers. We also used the nonlinear circuits to make physiological sensors. And then we combine them together to give us more information. So this kind of effort will rely on knowledge about the physiology, and the real-day biological questions. It will also rely on the detailed understanding of the physics, of the electro engineering, and about the mechanical engineering fabrication techniques. So I would say that this kind of project is very interdisciplinary, that will bring people from different backgrounds together. So instead of a topic that might require normal research, NMR is a very good way to send us new information, send us new message from its radio frequency. But at the end of the day, MRI still has the challenge, because we have to justify the cost and the relative non-portability of an MRI in real biological fields whose expectation often far exceed our capability. But as we work along our path, and while we are desperately searching for future destinations, maybe the scenery on the pathway itself is something that's most worthwhile to explore. So I would appreciate your feedback, and thank you for your attention. Thank you.